So today we have Lonike Faes here. Lonike is a PhD student, a fairly senior PhD student, I think maybe in the final year or so, working in Maastricht, in the group of Federico De Martino, which specializes in auditory cortex. I believe you will present an overview of kind of the entire PhD project, which I'm aware of three projects, and namely, initially we were looking at layerfMRI VASO in auditory cortex, which hadn't been done before, and we figured out why. It's then hard. Then after that, you specifically, or in parallel, you looked at Nordic denoising and its impact on kind of signal reductions and then how it affects beta scores. And then more recently, also a kind of quote unquote application of layer dependent fMRI to look at kind of mismatched negativity or kind of these paradigms. I forgot the right term for it, with and without, I think, deveining models. Please try sharing your screen, please.
Okay. Thank you, Renzo, for this introduction. Let's see. So you should be able to see my screen now. That I'm going to put, if I get the Zoom window out of the way, as a presentation. Can you all see the screen and hear me well?
Looks good.
Okay, great. Well, then thank you, Renzo, for this nice introduction. I think you've covered indeed everything. So my name is Lonike Faes, and I work at Maastricht University as a PhD student. And in the next 40 minutes or so, I'll give you an introduction into the projects that I've been doing during my PhD, which have all centered on using high-resolution fMRI or layer fMRI in the auditory cortex. And my general interests in terms of research focus on how our brains process information that we expect to hear, or stimuli that we don't expect to hear that deviate from our expectations. As Renzo mentioned, something related to mismatch negativity, but then with high resolution fMRI. And I find this very interesting because our environment is very dynamic, it's very complex, and especially in the auditory domain, information that reaches our ears is often very noisy and incomplete. And by anticipating what we will hear next, our brains can actually help us in prioritize behaviorally relevant information and, for instance, facilitate auditory stream segregation and prioritize behaviorally relevant sounds, such as speech in noisy environments. And a widely referenced theory that relates to these predictive processing that our brains perform is called predictive coding. And that is something that we would like to investigate. And it suggests that there is a process of active inference going on that happens through a hierarchical process of information exchange. So the idea is that we have a generative model of the world around us stored in our brains that can actively send predictions, feedback to lower order areas where it can be compared to sensory input. And this comparison, this difference between the two, is what we term a prediction error, which is what is being fed forward to update this internal model. And this is a reiterative process. So this is the third project that I will talk about today. And that is because before going into these neuroscience questions that we would like to answer using high resolution fMRI, we first wanted to investigate a few other things. And first of all, I think I assumed, as many of you here know, when we're doing high resolution fMRI, we always have two concepts in mind, namely sensitivity and specificity, because we want to have a high signal, but we also want to be sensitive to signals that are localized to specific cortical layers. So we want to be spatially specific in measuring areas that are close to the locus of neuronal activity. And many high-resolution studies that have been done have used gradient echo bolt acquisitions, while we know that they're more sensitive to macrovasculature, including large penetrating veins that drain blood from deeper layers to the surface. And this displaces our signal from where the activity is happening to the surface. And so we often have a bias in this signal. So alternative things have been suggested when it comes to improving specificity. And one thing that, for instance, in the auditory cortex has been used before is, for instance, 3D grays, which has been shown to be more spatially specific. But it was a bit limited in the coverage that Michel and Morel, for instance, that I put the reference below, has found. And with Renzo being in Maastricht at the moment at which we started this project, we thought it would be a very good idea to try and investigate whether we could use VASO to study the auditory cortex, as it's been shown to be more sensitive to the microvasculature. And so we thought it would be a great idea to investigate the feasibility of VASO. A second thing that we were interested in is related to methods that improve the sensitivity of our data. And the reason why we were specifically interested in this was because I am working on a very large NIH grant. And the initial purpose of this grant was to collect data with the same experimental paradigm in both healthy volunteers and in patients. And with patients, we're a bit more limited in the amount of time we can scan them. And so if we can collect less data in these participants, it would be great if we have some sort of a method that can improve our sensitivity when we only have a few runs that we can collect. And so I will also talk about this, that is Nordic. And then later I will circle back to the initial few points in this introduction. So the first thing that we wanted to investigate together with Renzo was whether we could use VASO in the auditory cortex. This study now has been published, I think, 2 years ago, almost in PLOS ONE. And as the title suggests, we looked at challenges and capabilities of using CBV-sensitive layer fMRI in the auditory cortex. And we divided this study into 3 sub-studies, as we called them. The first one was really about protocol development and artifacts that we found. And I actually today didn't want to focus on what exactly were the problems that we encountered, but more focus on the capabilities. And so I'll immediately move to the second part of the study. The second sub-study was the question whether we would use a 2D or a 3D EPI readout. This is stemming from discussions we had when we were at the scanner, where usually for auditory cortex fMRI, we have used a 2D EPI because there's a lot of physiological noise in the auditory cortex. And 3D EPI is a bit more susceptible to this from the experience of the auditory specialist, whereas we also had Renzo on the VASO specialism, who said, well, I've usually used 3D EPI, and then we thought, let's put this to the test. And then the third sub-study that we did was we wanted to show that we could also then apply VASO to study something related to frequency specificity in the auditory cortex. So the first thing that we did is we used a 30 seconds on, 30 seconds off, very strong auditory stimulation paradigm. So we blasted our participants with sounds. This stems from the fact that in our protocol development part, we initially had no activation whatsoever when we did present sounds. And so Renzo came up with the great idea of just blasting them with sounds. Multi-frequency sweeps, we called them. They were horrible for the participant to listen to, but with a protocol of 12 slices, 0.9 millimeter isotropic data, and both a 2D and a 3D sequence, we managed to get activation in the auditory cortex. So we're looking at this area here. And when we visually inspect these maps, we can see that we get nice activation in areas where we expect it, but a bit more in the 2D versus the 3D. So we collected in the same participants 2 or 3 runs of 2D and 3D. It was always the same number of runs per 2D and 3D. And especially when you look, for instance, at participant 4, we don't really have a lot of activation in 2D, but we do in 3D. So in terms of activation, this speaks towards using 3D. Then the other thing we tried was we drew a functional ROI and an anatomical ROI, and then looked at the response to 2D and 3D across the layers. In this case, we didn't really have an idea of what we would see. All we use this for is what method gives us the higher response, and this was 3D. So in the end, we decided to continue for the application of a tonotopic study. We changed the auditory stimulation to have a 23 seconds on, 23 seconds off auditory stimulation. And we presented 7 different center frequencies. And then per block, we present 1 frequency and then slightly alternate between along this center frequency. So you then don't get a very clear beep, but you get a nicer sound to listen to. And we use the sequence that we got above. And what we're looking for in these maps is they're maps of frequency specificity. So it's been shown before that you have an area that prefers low frequencies on Heschel's gyrus, and this is outlined in black. And it is surrounded by high frequency preferring regions. And this is visible in our participants. So we took this as a proof of concept that we can map tonotopy in the auditory cortex using VASO. We try to refrain really from showing any bold data in the paper itself. It's on supplementary materials, but to give you an illustration, in the bold data, we have a similar pattern, but it's less noisy than in the VASO data. So in short, yes, we can use VASO to image the auditory cortex, but it was quite challenging. So I didn't dive into all the details of the piloting session, but we did want to write this down in the paper. We can get bilateral coverage in all our participants. So that's really an upside compared to, if you compare it to gradient echo bold, where you also always get bilateral coverage, VASO is doing a very good job, but we're limited in how many slices we can actually get. The 12 slices at the 0.9 millimeter resolution is a bit limited compared to other methods. And for what I would like to use fMRI for, this may currently pose some limitations. So what I would like to look at is quite weak effects, because we want to present rare events. And at the moment, I would say with this paradigm, this would still be challenging if we want to use VASO. So who knows in the future, but for now, I think it's a very good starting point, and we will see where in the end this will take us. Then the second thing that we wanted to look at was related to the sensitivity. At the moment of investigating this, Lasse had not written his blog post yet about how to use or when to use Nordic on VASO data. So what I presented before is does not use any Nordic. Now, what is Nordic? In short, Nordic is a denoising technique that specifically targets the removal of thermal noise. So in our data at 7T with our voxel sizes, usually we are in the thermal noise dominated regime compared to physiological noise. And it does a principal component analysis, and it removes components that are indistinguishable from Gaussian distributed noise, which is the thermal noise, while leaving your components that contain signal intact. It's been shown that Nordic increases statistical values while leaving response estimates unaffected. And it's been tested already in a few other publications where people have looked at different areas, different experimental paradigms, resolutions, and field strength. And so we decided to give Nordic a go to see what the effect would be of Nordic on our high resolution gradient echo bold data. So the data we collected is, we have 10 participants, 0.8 millimeter isotropic voxels, 42 slices, and a TR of 1.6 seconds. And here we collected about 6 to 8 runs in each of our participants, and we presented our sounds in a slow event-related design. We collect 230 volumes plus noise scans, which is something that the algorithm can either use or ignore. And from this, we collected we created 3 data sets. So it's all the same data. The difference is in the first step of the processing, do we use Nordic or not? As we did in the original data, there we did not use Nordic. Do we use Nordic and the default settings? That means it uses this noise scan. That's a scan appended at the end without any radio frequency excitation. And then it uses a more empirical estimation of the noise level to determine which components are noise and which components are signal. Or we also created a version where we don't use this noise scan. We call this Nordic no noise. And then it uses a bit more of a theoretical estimation. And for us, the most important thing here is if you don't use the noise scan, we're a bit more conservative. So we were more thinking that we would see a grade in original Nordic no noise and Nordic default when it comes to our metrics that I will go into in a second. I will later also describe in more detail the experimental paradigm when I go into the thought behind this. So one more note before I continue. This experimental paradigm and this study was not really designed to evaluate Nordic. We had a purpose in mind that's related to predictions. So keep this in mind for later. So the first thing that we wanted to look at is what is the effect of Nordic on activation maps. So here I've put t-value maps where indeed we see that if we move from the original to the Nordic no noise and the Nordic default data, we can see an increase in number of active voxels and also in how high their t-values are. This is in agreement with previous studies, so it was more a replication in single subjects. However, when we then looked further into the response estimates, so the beta values calculated in % signal change, we can see a drop in beta values. So maybe I should add before, I will show you all sorts of plots that are across participants, but in the supplementary materials of this paper, also all the plots are available for single subjects. But we see a drop in beta values when we look at the betas before Nordic, the betas after Nordic default, and the betas after Nordic no noise. Yet we also see an increase in t-values in our regions of interest. So that indicates that the variance is reduced even more than the reduction in beta values, resulting in these increased t-values. And this is especially apparent in voxels that contain very low signal. So here, what we approximated here is the TSNR. So we calculated the mean of the pre-processed data and we divided this by the standard deviation. And here we can see that in general, betas tend to change when we use Nordic. So in the same voxels, we plot the beta before Nordic minus the beta after Nordic. So in both cases, the betas change, but it becomes less the higher the signal in the data. So especially in low TSNR voxels, we see a change in these betas, and probably this is where this reduction of betas is coming from. However, if we then look at the reliability of estimates, so we calculated this in split half correlation, we can see that especially in the areas where there is signal, meaning there are beta values, there is a higher % signal change, we can see that the reliability in these beta map estimates are increased. Comparatively, the t-values actually are more reliably estimated in most of our cases. And this is generally a trend we see among participants. So then the question arose, when we have this reduction in beta values, is it because there is part of the signal maybe that is being removed from our data? So we partitioned the variance in different parts. One is relating to how much variance in general there is in each data set, the variance in the data that is associated with the experimental paradigm, and finally also something that we termed the residuals after Nordic, that is basically our estimation of what is removed from the data when you use Nordic. And what we see here is that when you use Nordic, either version of the two, a higher proportion of the variance is explained by the design in our data. So this also speaks to what I said before. There is a reduction of the variance in your data, which I think is a normal consequence of this denoising method. You're taking noise out of your data, you're removing variance. However, if then we look at what is removed from our data, we here see that also a portion that is related to our design is in the variance that we remove from our data. So it seems that there is a bit of signal that we lose when using Nordic, more so than when we use the Nordic without the noise scan. And then finally, we explored what the effect would be of Nordic on our laminar plots. And I've plotted here, again, across participants, the laminar response to 2 of our conditions. And here we see that this beta reduction is most pronounced at the surface. And this is replicable, at least in our data set, across participants and across conditions. So what are the conclusions that we drew from this study? Nordic improves your statistical values. We see increased t-values. And especially in areas where there is some signal present, there is some indication of increased reliability in response estimates in these beta values, but also in the t-values. Though we do have this reduction in beta values that is present across our participants. And this may be especially related to low TSNR voxels. And so that brings us to the point that when we do Nordic, in our case, there is some signal that is being removed from our data that is indistinguishable from noise. It could be related to the fact that we have a slow event-related design with a very small effect. So we have low power. But this reduced magnitude of the effect, so our reduced betas and an increased inter-subject variability after using Nordic made us a bit more hesitant to use it for the next study. So in the end, we decided, also given that we don't have patient data in the next study for which we really need an increase in sensitivity, we decided not to continue with Nordic, at least for now. Which brings us to the final part that I wanted to present today, which is about the predictive processing using pure tones. I'm mentioning pure tones because there is another project in the pipeline that uses syllables. And for this, we then use the original data set that I described before. And here we want to understand how the brain processes expectation violations or stimuli with a weaker predictive context, which I'll get to in a second. And as a little reminder, what we want to understand is how feed-forward and feedback processes happen. So do we see evidence of predictions or prediction errors? We have a 0.8 millimeter isotropic voxels and 42 slices, which allows us to cover 5 areas in the temporal lobe. And this we thought would be interesting to image, to look at 5 different areas, as then maybe at some point we can look into a hierarchy along the auditory cortex. And in this case, we used gradient echo bolt data. And as Renzo mentioned before, we've also adapted a model that is described in the 2 papers that I put at the bottom that take into consideration the draining vein that I talked about in the introduction. So first, to give you a little background on the experimental design, our participants passively listened to tone sequences presented in a slow event-related paradigm. And what was common to all of these tone sequences is that they always consisted of 4 tones. So in the case of the predictable condition, for instance, the ordering of the first 3 tones, we call them contextual tones, elicit a prediction of what the 4th tone would be. So if you hear ascending frequencies, then your expectation would be that the 4th tone would then still be at a higher frequency. And we have both an ascending and a descending version of this. But in some cases, we would violate this prediction. So when you have the expectation that you would hear a low tone, we then present a high tone. And this would elicit an error. We call this a local error. So because in this sequence, you present a local in this trial error. And we also had another type of condition where we scrambled the order of the first 3 tones to reduce this prediction. So if you scramble the order, you don't have this clear ascending pattern. And so you would not have such a strong prediction of what the 4th tone would be. Moreover, we manipulated, in addition to this local violation of a pattern, also the rarity or global pattern of how often we will present each of these sequences. So the predictable condition we would present 10 times per run, and the mispredicted and unpredictable conditions only 4 times per run. So that really you would expect to hear this predictable condition because it's much more often presented within the experiment. So we collected functional data. So it was a 2D multiband sequence with slice scan time correction, motion correction, temporal filtering, and we did distortion correction. And to use the model that I'll describe in a second, we also had to pre-process our anatomical data. So here I've put an illustration of the high resolution MP2 range image that we collected with on top the coverage of our functional data slab. And then here would be the area that we're interested in, so we can cover quite a big part of it. And from this MP2 range image, we then create a high resolution segmentation, which I've manually altered to make sure that the gray matter, white matter, and gray matter CSF boundaries are accurately delineated. I draw 5 regions of interest that you've also already seen in the Nordic paper, but now illustrated on a surface. And per ROI, we can use the equivolume principle to then layer the specific region of interest. And then for the modeling, we selected voxels that were responsive to auditory stimulation. So that means that we took a threshold of t is equal to 2, and the voxels that were above this threshold, we selected for our further analysis. And then we used the functional and anatomical data to then put this all through a model. And I want to give a little thanks to Isma here, who has been of invaluable help on getting this part of the project up and running. Isma was a former postdoc in our lab, and she's really has done great work in making sure that we can use this model and that we know how to implement our data to infer neuronal population parameters. So I've put a schematic here. It's based on what was put in the original publication of Camille and Martin, and it consists of several parts. So we have a neuronal model that is consisting of 3 depths, and in each depth we have an excitatory and inhibitory neuronal population. And through neurovascular coupling, it's linked to a number of vascular depths. And here you have something that takes the microvasculature into account, and also here this ascending veins. And this is the main reason why we use this model, to take out these ascending veins. And if you use the forward way of this model, you can estimate laminar bolt responses. But through model inversion, you can also infer hidden neuronal parameters when you have the measured laminar bolt responses. So that is what we used it for. When you then look at this little bigger schematic of the neuronal model, you can see that we specify 2 types of input. We have a driving input and a modulatory input at these 3 neuronal depths. So the driving input in our case is the auditory stimulation. So we have we want to compare 2 different conditions, the predictable and the mispredicted for now. This driving input is the auditory stimulation. So we always, in both of conditions, present an auditory sequence that consists of 4 tones. You expect a response in general in our regions of interest to this auditory stimulation. And then we add also a modulatory input, specifically to the parameter that then causes a modulation of the shape and amplitude of the bolt response. So what we're trying to infer is how the bolt response changes shape when we present this violation of the prediction. So you expect that if you present auditory stimulation, there is a certain driving input, there's a certain bolt response, and then you have an additional modulation that is very much related to how it changes when you present a violation of this expectation. Our null hypothesis would then be that our response is the same, so there's no difference between the predictable and the mispredicted conditions, but we create a model space of these 8 different models where we give as hypotheses in which of the layers we would have this modulatory input. Is it only in the superficial layers? Is it only in the deep? Or is it any combination? Or is it in none of the layers? And this is then the value that we plot here. So in this case, a modulation of 0 means that there is no difference in this response between the mispredicted and the predictable stimuli. And any positive response means that we have a higher response to this mispredicted stimulus compared to the predictable ones. And when we look at these results and we try to implement this in the architecture of predictive coding, we find an error signal located in planum polare and a middle response in posterior STG. Based on this architecture, this suggests a routing from the planum polare to a higher order area that is posterior STG. Interestingly, we also found a deep layer effect in all of our regions of interest that is not related to prediction, but it is in line with animal electrophysiology that use a very similar paradigm. So the idea that they postulate that is in line with our data is that you have an early error component that is related to the error within the little sequence, a late error component that is related to the regularity with how often you present a certain sequence within this experiment. And then they find a late feedback component that they interpreted as a global model update. So when you have errors, they need to be incorporated in the model. And this is what they saw in a feedback component, which could be the same as what we find in our deep layers here. When we then look at the unpredictable condition, we expected here that if you have a weaker predictive context, you would not really have an error. And so we also don't see this in any of the superficial or middle layers. But interestingly, we still see this deep layer effect in some of our areas. And we're thinking that this could still be related to this global model update, because in the unpredictable conditions, we still present these conditions less often than the predictable conditions. However, we also think that this is a bit less lower powered than the mispredicted condition. And so it's here we found evidence of significance in 3 of the areas. And things to test in the future. We could, for instance, in the model, incorporate connections between layers and between areas to test these hypotheses related to the routing of information exchange. And another thing that we could test is to make our experimental paradigm more similar to what has been shown using animal electrophysiology. So they used a similar paradigm, but not exactly the same. And so if you want to test the hypothesis that this deep layer effect is indeed a model update, then we could think of a paradigm that is more similar to what has been used in these animal studies. And so that is something that we can try in the future. And then finally, what is coming up next, though I don't think this I will manage during my PhD anymore, is we have a similar paradigm now as what I discussed before, but then using syllables. And in this data set, we now have 10 healthy volunteers and 3 patients. And we also have some ECOG data that is collected in these 3 patients. And so they have done the exact same experimental paradigm in the scanner as what they did when they had ECOG. And it would be great to see how we can combine all of this together with more temporal and more spatial information to see whether we can, for instance, already test certain hypotheses that I described above. And another thing that we could do is, given some caveats in the experimental paradigm that I discussed before, and to make it more similar to what has been done in animal electrophysiology, we're currently piloting a new data set. I have 1 volunteer at the 10.5, so collected in Minneapolis, where we changed the design to be more similar to animal electrophysiology. and to help us disentangle certain errors that we couldn't disentangle in our previous study. And with that, I would like to thank all the people that have been involved in these projects. And I think I'm quite well on time. So do you have any questions?
Thank you very much. Thanks for this nice overview of quite a lot of work. Maybe I missed it now at the end, but did you show us a feed-forward condition for the mismatch negativity?
Oh, sorry.
Kind of you showed the model outputs for the random, like the unpredictable condition, and also the kind of unexpected, like the mismatch negativity, but not the feed-forward one. So I was wondering if the like model then this is the mispredicted one, right?
Yes.
So in the model for the 60% of all the trials, you used a kind of predicted sequence of tones. How does the model perform with that? So does it show the middle feed-forward layer, or kind of does it take care of the veins, or?
Yeah. So what it does is it always looks at two conditions at the same time. So there's not a separate feed-forward condition, but we do give it the driving input, which then would be the auditory stimulation. But it doesn't give you as an output that it's like the middle layer that is active if you hear something compared to rest, or compared to not, compared to rest. But what it does is there's auditory stimulation in both conditions. So it first tries to fit whatever is common to both, which is the auditory stimulation of 1.6 seconds. And then it tries to fit, okay, what is actually then changing in this bolt response that is then different compared to the other predictable condition.
Gotcha. So it's kind of in your model, you had these big arrows, and then the little arrows, the red ones. It can only look at the red arrows. So you need to have three conditions, like two. Right.
Yeah. Yeah. So it first fits this part, and then it fits this part. But then indeed, what we eventually get as output and what we look at is then what I've here put as the red errors.
Did you or Isma have to kind of update it? Like the model originally, I think was, well, like at least the previous deveining models from Irati or other folks and Martin, they kind of was optimizing it and validating it on primary visual cortex with its kind of unique angio architecture. Did you have to update it based on the angio architecture of the auditory cortex, which might be slightly different as Farouk showed, or is it kind of fitting that too?
It's fitting not just these parameters, but we did make it the response, the slope of the how fast the bolt response increases, that we did change to a shorter time. So it's shown that in the auditory cortex, the bolt response is faster than in the visual cortex. So this we did change. And then the other things mainly are fitted, the parameters. So yeah, I didn't bring a table with the other parameters that were fitted. But so yeah, we increased the speed at which the bolt response increases. And other than that, we did not really adapt anything specifically for the auditory cortex.
You didn't look at the beta scores, right? That you could have also kind of analyzed those paradigms on. Like it is a relatively common and simple paradigm, I believe, for layer fMRI, where you have kind of two conditions and you could see how the bolt signal changes for predicted versus unpredicted, or you didn't look at the betas, did you?
I didn't look at beta values. We kept it similar to what we got in what the model takes as input. So it works on event-related averages. And so what we did look at is if we take the maximum response in these time event-related averages, how does the data look without the modeling? And what we can see, first of all, is we have a nice draining vein effect, which is what we expect and also what we use the modeling for. And what we've noticed when we don't use the model is that we have a lot more significance, especially for the mispredicted versus predictable. I don't know how well you can see. but it's much more. So blue is data only. And we can see here a lot of significant values. So our model in this regards is not inducing any significance. It's actually reducing how many significant effects we have. And this is especially apparent in the middle and superficial layers where you expect the modeling to reduce the draining vein effect. So because we can, of course, look at the difference between conditions as people would do when they use subtraction in laminar fMRI. We've done that. But when we use the model, we see especially at the middle and especially at superficial layers, a reduced difference between the, an increased difference between conditions.
It looks like this deep layer effect is not an artifact of the model, right? You have it to begin with.
Yeah.
Maybe if nobody else has a question just yet, I have a question about Nordic. So you had these kind of with and without noise scans, which is then, I believe, based on Federico, they kind of changed the algorithm in the MATLAB version that's shared by fMRI, that when you're not using the noise scans, it's kind of more conservative. It's basically like Nordic Light. So I'm wondering, based on this, like the results that you're seeing are results of the kind of Nordic MATLAB version that is shared, which might be kind of a result of the feedback that it was reducing too much the betas when you're not having the noise scans. Or like you understand my struggle to figure out the circularity. So is it a feature of Nordic that when you're not using the noise scans, it's kind of a Nordic Light, or is it kind of basically tuned that way so you're not having the beta scores so much? Like as far as I understand your data, if you're not using the noise scans, the DSNR boost and the statistical value boost is not as big, but also the drop in the beta scores is not as bad. So it is, and that's why I'm calling it Nordic Light, maybe.
Yeah.
Which might have been a design choice in the algorithm based on initial feedback by looking at the beta scores. Is that right?
So there's no one in the audience, I think, that really is about Nordic development. I'm not so sure. As far as I know, this all is done on the latest version of the scripts that Steen put online in 2021 or something. So in that case, it's not Nordic Light is not something that is added to reduce this effect. As far as I know, this was part of it because not everyone can easily collect a noise scan at the end, or especially in the beginning. I think you also struggled initially with when do I collect a noise scan? It needs to be appended to the run. And I think they just added something where then you don't estimate from your data anything related to the noise level from this noise scan, but you can take some other already established theoretical threshold. So if you, I mean, you would have to check with Luca, but as far as I know, in terms of circularity, this paper is then based on the latest scripts.
Gotcha.
And then
yes. Like you're saying that the drop in beta scores when you use Nordic is mostly in voxels that have low SNR. In your layer profiles, you don't see a bias there though, right? Or like maybe it depends on the protocol exactly. Like if you're more limited in the superficial layers by CSF pulsation, physiological noise, as opposed to deepest layers, which might be more thermal limited. But overall, it didn't screw up your kind of your layer profiles, right? It was just a scaled version of the original no Nordic. It was just kind of reduced.
Yeah. So we also argued in the paper that we could look at differences between conditions before and after Nordic, but then we found this challenging as we don't know what the ground truth is, because we also have a very weak experimental paradigm. So it would be good to test maybe something a little bit stronger and to see then how this difference in slope changes the experimental paradigm. But yeah, within single subjects, I would say you see a similar type of decrease in your slope of your laminar plots.
What do you mean by not knowing the ground truth? Like averaging a lot of people and runs is usually considered the ground truth. You didn't trust that one when doing Nordic as a
I
ground truth?
mean, so what we found with other things is that it does increase the variability across subjects. So it seemed to be that in our case, in some of our metrics, Nordic was improving in some participants, the stability, but not improving this in other participants.
Okay.
So we had a lot of unknowns of what exactly is going on.
Understand. So in both of the Nordic versions that you applied, the with and without noise scans, you
Mm-hmm.
always use the complex valued one, right? Where you have the magnitude and phase. Is that right?
Yeah.
Okay. Is there anybody else who has a question? I think you answered all of my questions too. So thanks a lot. And I will put this on YouTube soon. Maybe for the next few weeks of this seminar, I will be in Austria for the rest of December and come back for in the second week of January. And then I think it might make sense to reshift the day of time and also the day of the week and then the time of day for the seminar. So I will reach out about a doodle or something. So maybe when we restart this in the new year in the second half of January, we reconvene at a different time because it overlaps with free time that we have on the scanner, and also for Peter, it's not always convenient. All right. So I wish you all a happy Thanksgiving. Thank you, Lonike, for this nice presentation, and see you all in the new year.